At the very heart of molecular biology, you'll find two superstar molecules, DNA and RNA. One of them holds all the genetic information that makes us who we are, while the other is in charge of actually using that information to build and run the show. So let's dive into how this incredible partnership works.
Here's a great way to think about it. Imagine DNA is the master blueprint for a massive skyscraper. It's incredibly precious, so it's kept locked away safely in the head architect's office. Then you have RNA. That's the busy construction crew out on the site. They take copies of the blueprint and get to work, actually building the skyscraper. It's this perfect dance between the plan and the builders that powers every single cell.
So let's start with that blueprint, DNA. Its entire structure, its whole reason for being, is designed for one critical job, to safely store all of an organism's genetic information for the long haul.
The full name is a bit of a mouthful, deoxyribonucleic acid, or just DNA for short. This long, chain-like molecule is basically the ultimate biological hard drive, passing information from one generation to the next. But what exactly makes it so good at its job?
Well, DNA's incredible stability comes right from its famous structure. That double helix shape acts like a twisted ladder, protecting the genetic code, the rungs, on the inside. And those rungs have strict pairing rules. A always pairs with T, and G always pairs with C. This is a brilliant design because it means you have a built-in backup. If one side gets damaged, the cell can use the other side as a perfect template to fix it. It's a genius system for reliable, lifelong information storage.
All right, so we have our master blueprint. Now let's meet the construction crew, RNA. If DNA is all about stability and storage, RNA is built for action and versatility. It's the molecule that actually gets things done. Ribonucleic acid, or RNA, is usually single-stranded. Now that might sound less robust, but it's actually its superpower. Being single-stranded lets RNA fold into all sorts of complex shapes. This means it can do a bunch of different jobs. It can carry messages, sure, but it can also become part of a cell's machinery or even act like an enzyme to speed up chemical reactions.
When you put them side by side, the key differences are surprisingly simple, but they have huge consequences. RNA uses a slightly different sugar called ribose, and instead of the base thymine, or T, it uses a very similar base called uracil, or U. Those small chemical tweaks, plus being single-stranded, completely change its personality and its job in the cell.
This quote really nails it. That one tiny difference in the sugar, deoxyribose, is what makes the DNA backbone so much more stable than RNA's. It's exactly why DNA is the perfect molecule for permanent storage, while RNA is much better suited for temporary, get-it-done kind of tasks.
Now, here's a key detail. We can't just talk about RNA as if it's one thing. It's not. It's much more like a specialist team, a coordinated crew where every member has a very specific and very important job to do.
The main players in this workforce are messenger RNA, the courier, transfer RNA, the adapter, and ribosomal RNA, which is basically the factory itself. Let's see how this team works in concert.
Okay, so first on the scene is messenger RNA, or mRNA. This is the courier. Its job is to go into the nucleus, make a temporary copy of a gene's instructions from the DNA blueprint, and then carry that message out to the main part of the cell. Once that mRNA message arrives at the construction site, transfer RNA, or tRNA, steps in. This molecule is the essential adapter. It reads the genetic code on the mRNA and brings over the exact amino acid that matches that code. It's literally translating the language of genes into the language of proteins.
And where does all this happen? Inside a molecular factory built from ribosomal RNA, or rRNA. But get this, rRNA isn't just the scaffolding for the factory, it's the actual machinery, the part that links all those amino acids together, forging a new protein chain. It's both the structure and the worker.
So we've got the blueprint, and we've met the specialized crew. Now let's put it all together and see how this incredible flow of genetic information actually builds life.
This table just lays it all out so clearly. Every single difference you see here, the number of strands, the type of sugar, the bases, it all points back to that one central theme. DNA is built for permanent, safe storage inside the nucleus. RNA is designed for temporary, active work out in the bustling cytoplasm of the cell.
And this entire process, this flow of information from DNA to RNA and finally to protein, well, it's so fundamental to every living thing we know of that it has a very grand name, the central dogma of molecular biology. from the permanent blueprint to the temporary copy to the final functional machine.
Now, this is way more than just textbook stuff. Understanding this flow, especially the RNA part, has blown the doors open for one of the most exciting fields today, bioinformatics. You see, by reading a cell's RNA, we can get a dynamic, real-time snapshot of what that cell is actually doing right now.
In a field called transcriptomics, scientists can analyze all of the RNA messages in a cell at once. Techniques like RNA sequencing let them measure exactly which genes are switched on or off and how loudly. This gives us an incredible picture of how cells function and, more importantly, how they malfunction in disease. It's like listening in on the construction crew to see which parts of the blueprint are being worked on this very second.
So let's circle back to our core idea, because it really is that elegant and simple. DNA stores the master blueprint. RNA takes that plan and executes it, building and maintaining everything the cell needs. One stores the information, the other puts that information into action.
And this leaves us with a pretty incredible question to think about. As our technology for reading and understanding RNA gets even better, what complex diseases, from cancer to Alzheimer's, could we finally get ahead of simply by understanding what our cells are truly up to in real time? The possibilities are amazing.